Thank you for your overwhelming response in 2020. We're continuing with our web seminar series. Today is our lecture number 19, and our guest speaker today is Dr. Vinod Scaria, all the way from India. But before we start the introductions, just a reminder to the audience, we're going to use the similar format as before. Please post your questions on the question answer tab on Zoom. Over to you, Rob, to introduce our guest panelists for this talk.
Thank you, Mohit, and once again, welcome to everybody. Happy New Year. We trust that it's going to be a stressful few weeks for us and our healthcare system, I think, but we wish everybody best wishes for health and safety as we proceed. Welcome to the new 2021 series. It's a particular delight to have Dr. Scaria with us today, and I'm going to pass it over to our two guest panelists, Vinod Chandran and Igor Jurisica, and Vinod Chandran will do the introduction for us. Thank you, Vinod.
Thank you, Rob. I hope everybody can hear me well. I'll assure all of you that I'm still on Earth, and the spaceship that you're hearing in the background is just a problem with my computer. I'd like to not waste any time and begin this year with introducing Vinod Scaria. Vinod, I know him for a long time. He trained in medical school that I came from, Calicut Medical College in the south of India. And after his medical degree, he saw the light and decided that he wants to spend more time in computational biology, and subsequently did his PhD at the University of Pune in India. And he's thus a clinician as well as a computational biologist, and his research interests are in understanding the function, organization, and regulation of the vertebrate genome and how genomic variations could potentially impact them. He's a senior scientist at the CSIR Institute of Genomics and Integrative Biology in New Delhi, India, and has done a lot of pioneering work in genomics in India. As you know, most of the genomic research across the world is primarily done on Europeans and more recently in the Chinese. And so Vinod is leading the research among other populations, especially South Asia and the Middle East. With that, I won't spend only one other connection that I'd like to highlight is Vinod's wife is a rheumatologist, and so that is, as linked to rheumatology here. So with that, I'll hand over the floor to Vinod, who will be speaking on translational genomics. Thank you, Vinod.
Thank you, Dr. Inman and Dr. Kapoor, and thank you, Dr. Chandran, for the invitation and opportunity to talk here. A good morning to all of you, and a Happy New Year ahead. Before I start my talk, let me do some mandatory disclosures because we do a lot of work with companies across the world in terms of tech transfers as well as consultancy. And I'll also want to start with a brief overview of what we do. We call ourselves as genome informaticians. That essentially means that we look on the genomes using computers. A variety of things that we try to do using computers and looking at genomic information. About a third of my lab works in looking at epigenetics and non-coding RNAs. So we work on long non-coding RNAs and circular RNAs. Almost half of my lab works at the interface of clinical medicine and genomics, where we largely try to understand Mendelian genetic diseases, and that's essentially what I'm going to talk to you today. And a very small part of my lab works in the area of pathogen genomics, where we try to look at pathogen genomes and also the host-pathogen interactions. Now, we were very fortunate to be in the right time and the right place. And this is a quite old slide, and I'm sure you have seen this over and over again. But just to put into perspective, we started our lab way back in 2007, 2008. And that was also the most exciting period in time because, as you see here, the cost of genome sequencing was dwindling very rapidly, almost exponentially. And as a consequence, a large number of human genomes were getting sequenced. Now, if you ask this question, at that point of time, were sequences uniformly distributed across the world? The answer was probably no, and it still is a no. And this is a slide from 2008. I still keep showing it. So what you see here is essentially the human genome diversity map from the Human Genome Diversity Program. And in perspective, I've put the whole genome sequences that are available at that point of time in 2008. And as you see here, the 6 genomes that were available at that point of time, this includes the reference genome, the Craig Venter and the Watson genomes, and the Chinese, Japanese, and the Korean genomes, which came in quick succession, were all from quite homogeneous populations. And what you see in the middle of this slide is a quite large and diverse and admixed population, which runs from Middle East to Central and Southeast Asia, almost encompassing a third of the world population. And what you didn't see there was essentially human genomic representation. And that's essentially the point where we started. And of course, we started putting together the pieces of the puzzle, learning the tricks of the trade. And I'm sure this machine most of you would not have seen. This is the Solexa machine, one of the first next generation sequencing machines available. And the idea was to essentially learn how to put together a genome right from assembly to looking at genetic variations and to be able to put them into the clinical perspective in terms of looking at functional genetic variations. And in late 2009, we announced the first genome from India, making it the 6th country to be doing genome sequences. Now, what we really didn't stop there, and actually we went across the region to sequence more genomes. This includes the first Sri Lankan, the first Malaysian, the first Philippines. And of course, we were one of the founding members of the Pan-Asian Population Genomics Initiative, which is a flagship program of the Human Genome Organization. And we today hold quite large databases of population genomes at IGIB. This includes the Almina for Middle Eastern population, SAGE for Southeast Asian populations, and quite recently Indigen, which was concluded last year for the Indian population scale genomes. Now you would ask this question, of course you could do human genomes and you could do it quite cheap today and quite fast in fact, but then what's the use of doing all these genomes? And the real answer to that is to actually put it back into clinical perspective. Now, if you want to put it back into clinical perspective, you need to get back to identifying the so-called monsters and sinners. And I'm sure as children, you would have read fables where you have monsters and sinners described in text and in many ways pictorial representations. Well, as you grow up, we tend to realize that there are no monsters out there, but essentially there could be genetic diseases. And in many ways, these genetic diseases could be because of very specific genetic defects in the genome. Now, put a number to this perspective, while they're called as rare genetic diseases, in fact, they're not really rare. Well, put together, there are around 3,000 odd genetic diseases documented in literature. And to put a number to this 3,000 odd genetic diseases, there are around 70 million people in India and around 300 million people worldwide. Now, the real problem with some conditions is that patients typically go through multiple clinician visits, multiple rounds of misdiagnosis, and spend considerable amount of time, effort, and of course money into the disease and being able to get to a diagnosis. And that's understandable because individually these conditions are extremely rare. Most clinicians would not have even heard about these conditions unless they're very prevalent and well described in textbooks. And of course, in many countries, including India, there is no comprehensive system for screening and diagnosis, or even in many ways prevention of these genetic diseases. And therefore, that is an essential problem that we need to start addressing. But of course, you could always look at the problems and always also look at the opportunities. And the opportunity in a country like India, compared to a lot of countries otherwise, is that we are a very large country. We have close to 1 billion population, that's around a sixth of the world. Our genetic pool is quite diverse, unlike most of the Western nations, and encompasses small and endogamous groups. Small endogamous groups essentially means people who marry between individuals of the same community, in many ways the same surname, and a small proportion of these groups are also consanguineous in nature. And even more important than all of these is that we still have a close-knit family structure. And in many ways, individuals can trace, if not 3, 4, or 5 generations, 5 generations of their ancestry. And practically everything in genomics starts with looking at the ancestry and drawing an extended pedigree. Now, of course, when we talk about genetic diseases, it's also important to keep in mind that there's a variety or spectrum of genetic diseases, right from common diseases. I'm sure you're all aware about this because the clinicians, that's what comes to your clinics. They're typically affected by common variants of very small effect sizes. On the other end of the spectrum are rare genetic diseases, typical examples like sickle cell anemia or hemophilia, which are caused by rare genetic variants. And in many ways, the effect sizes are very large. Or in other words, they are predicted. Intermediate to them are essentially common diseases with rare variants. In many cases, they're actually missed in the diagnosis just because of the fact that they're essentially common diseases which are Mendelian in nature, but nevertheless not really genetically surveyed or diagnosed. So the tools and methods to look at these genetic disorders are also quite different. As clinicians, you would have all heard about genome-wide association studies. What we look at is the other end of the spectrum, where we look at exome or a whole genome or a gene sequencing, because we are essentially looking at extremely rare genetic events. So way back in 2015, with my colleague and collaborator, Dr. Sridhar, we put together this consortium. And the idea of the consortium was quite distinctly different. That we will work with clinicians across the country and try to help them rather than trying to publish more papers. And the idea of this consortium was that whenever they come across a family with an undiagnosed disease or where they have a clinical suspicion of a rare genetic disease, they could refer to the program. And we would do a standard way of consenting, genome sequencing, analysis, and reporting. And whenever we could solve a new genetic condition or a new gene or a new genetic variant, we could also take it up for in-depth biological studies and eventually get back to the clinician with a report so that he can act upon it. And over 5 years, today we have close to around 280 clinical collaborators from around 70 major clinical and research centers across the country, making it one of the largest rare disease genomics programs, probably across the world, if not entirety of Asia. And the idea is not just to do research, but also to improve education, awareness, implement diagnosis, and of course, finally, to implement policy, because there's a lot of policy that needs to change, not just to intervene in terms of screening and diagnosis, but also in terms of treatment, because a large number of conditions are today treatable. And behind all of this is an informatics system. I will not delve deep into it, but I spent time trying to explain probably 3 or 4 cases to just exemplify how this system works. Now, this is one of the first families that we got referred from the D.Y. Patil Hospital in Pune. I'm sure there are not dermatologists in here, but this is a clear-cut genetic diagnosis of lamellichtiosis. 2 children born with lamellichtiosis is something what we consider as criminal offense, because this is a condition which is genetic. At least a genetic counseling should have been offered to these parents, which was incidentally not offered. But to make things worse, there was actually improper diagnosis. Somebody had actually prescribed a karyotyping for this family. And as you see here, the karyotype showed an abnormality, which is also shared with one of the parents. And as you see here, the parent's skin is normal. So this is essentially the problem of genetic diseases in the sense that awareness is not really great. So you end up either misdiagnosing the condition or even offering them a wrong diagnosis for a condition, in many ways not even offering them what is very simple, which is genetic counseling. Now, typically when such families come to us, we do an exome sequencing for the family. And what we found was a genetic mutation in the TBM1 gene, diagnosing this condition as lamellichtiosis. We also do validation with sample sequencing to sort of prove the condition, and not just prove the condition, because almost all of these families would come back for genetic counseling, and you would need to offer them a genetic test, in many ways a prenatal test that will help these families. Now, this family actually opted for prenatal testing, and a 3rd child was born to this family, as you see here, with perfectly normal skin. Now, to make things much easier for this family, this genetic condition, when it was diagnosed, there's a lot of literature which suggests that this condition could be treated with retinoic acid, low dose. And these kids were put on low dose retinoic acid, and as you see here, the skin has practically cleared up. They start growing hair, and if they are fully clothed, nobody would ever recognize that even they have a genetic disorder. In other words, this is a happy family today with 3 normal children, right? 2 who have a genetic disease who could be treated, and a 3rd child who is perfectly normal but devoid of the genetic condition. This is another family from a place called Agra. That's where the Taj Mahal is. And this family was in news way back in 2015 because the parents had petitioned the President of India to euthanize all of their 6 kids, 6 of their 8 kids rather. And as you see here, the kids from the younger to the older one had a genetic condition, quite progressive neurological condition, which makes them bedridden. Now, just think of the plight of a family who is asking to petition, who is asking the President of India to euthanize their own children. And that's largely because they do not have a diagnosis for this condition, nor a treatment for this condition. Well, we ended up doing a whole exome sequencing for this family. We did make a diagnosis of MLC1 or megalencephalic leukoencephalopathy with subcortical cyst type 1. And what is very peculiar to this family was an extended pedigree was drawn. And as you see here, it's a quite consanguineous family pedigree. Just by drawing a family pedigree, we identified another family with another child with the same genetic disorder, who was also tested positive for the same genetic mutation. We did an extended family survey, the community survey across this small population of around 100 individuals staying in Agra. And what we found was quite remarkable. Around 28% of individuals were carriers for this extremely rare genetic mutation, which was described in literature only once before. And that really opened our eyes because such a large carrier frequency is possible only, of course, if you have a consanguineous marriage, which is, as you see here, in the pedigree. But what is really more important is that this community has a founder population originating from the Middle East, and today encompasses around 50,000 individuals spread across 3 states. So what's really more important is to not really change the practice of consanguinity, because that is something what is tradition, but actually offer them a solution to the problem, which is to offer them carrier screening and prenatal testing so that they can actually have normal children born out of their marriages. And to put in perspective the timeline, you could see that right from the first case referral in 2016, we have in 2019 a diagnostic which is approved and in the market, making it one of the fastest diagnostics which is now widely used for the population for screening, because this is possible with awareness and by reducing the cost of testing, and also making it widely available for individuals in the community. We also found another 2 families with individuals with the same genetic condition who could now be screened and diagnosed for this extremely rare genetic condition. I'll shift gears and, of course, tell you a story. And just to put in perspective, there is something called the malady of the Brugenzas. Now, for historians out there, this is a curse to King John of Portugal, who apparently kicked a monk while begging for alms. And the monk cast a curse saying that you would never have a first-born male in the family to live long enough to reach the throne. And the curse was so specific that the historians say that even illegitimate children to the family who were male didn't survive long enough. Now you think that's in Portugal, but this is a family right here in India. And as you see here, there are multiple male deaths, and male deaths out of pneumonia or sepsis. And if you're in pediatrics or you've ever been in the pediatrics ICU, you would realize that neonatal pneumonia or infantile pneumonia and sepsis is one of the commonest causes of death and well-documented cause of death across, not just in India, but probably across the world. Now, what was really suspicious was at that point of admission, 2 children who were admitted at the same point of time were apparently related to each other, and that raised a suspicion that this was something very genetic. So that's how we drew the pedigree. And for people who are interested in immunological disorders, a male child with pneumonia or sepsis, the first thing that you need to rule out is essentially B-cell deficiency, a condition called X-linked agammaglobulinemia, especially segregating in the male members of the family. And so therefore the B cell counts and the IG profile was, hemoglobin profiling was done. And as you see here, the B cell counts are almost nil with a very low immunoglobulin profile, sort of making the diagnosis of X-linked gammaglobulinemia. So typically what you do for such a condition, you need to go and find the mutation. So we did an exome sequencing for all 3 individuals and we found nothing. And that was very surprising to us because we thought exome sequencing would solve every single problem. But nevertheless, we got nothing in the standard analysis. So what we typically do for such condition is to actually attempt doing a whole genome sequencing. We did a whole genome sequencing, and to a surprise, we found nothing again. Now, that was really surprising to us because we were very confident that this is a familial condition. We have 3 members of the family. We could look at segregation, but nevertheless, we didn't find anything. So we were probably looking at something that we miss in standard analysis, and this is something that is missed across the world, not just in our lab. There are structural variations. So we went back and looked at structural variations, and what we found was something very remarkable. We found a very large deletion in the BTK gene causing X-linked gammaglobulinemia. Now what do you do? Since it's a large family, you quickly go back and make a diagnostic, and you could make diagnostics using MLPA, or multiplex ligation dependent probe amplification, a very specific assay to look at very specific chromosomal deletions. You could sort of standardize this assay and roll out to the extended family, because practically every member in this family, especially females, would benefit from carrier screening. Of course, one of the children succumbed to the infection, but we could save the second child because the child could access hematopoietic stem cell transplant very early in time. And now we have close to 19 members in this family who could now be screened and counseled, and in many ways, an early diagnostic test to be implemented, because if you can diagnose them at the newborn state, you could probably sort of triage them for hematopoietic stem cell transplantation, and that could practically save these children. And now HSCT is not really the treatment of choice for such children. In most of the Western countries, immunoglobulins are provided. But of course, in a country like India, long-term immunoglobulins is something that's unsustainable. In many ways, the families cannot afford it. But a one-time treatment of hematopoietic stem cell transplants are actually afforded by the government as a one-time cost. So most of these patients do undergo HSCT, and that's something very unique in a country like India. Now, from this one single large family, today we have around 350 odd referrals from this one single hospital. That is Government Medical College in Calicut. That's something that connects Dr. Vinod Chandran and me. And today we have a very specific program for pediatric immune disorders, where we offer not just diagnosis and treatment, but also patient education, and of course, a whole gamut of policy implementation in the state of Kerala. Now we've extended this across the country with multiple centers in multiple different states, including Delhi, Gujarat, Pune, Kerala, Tamil Nadu, and Puducherry, extending the reach of primary immunodeficiency disorders. We don't call them primary immunodeficiency disorders anymore, but rather call them the inborn errors of immunity. Now, this is something that is very close to us, and where we have really built a very large cohort across the country. And that's when we realized that immunodeficiency per se is just one part of the entire puzzle. Inborn errors of immunity today encompass close to around 400 genes or genetic disorders, making it one of the largest classes of genetic disorders known to humankind. Now, the spectrum of clinical presentations could actually vary depending upon what are the diseases that you have and what are the genetic defects that you have. Immunodeficiency, of course, is something that is very important because that is something that can present as recurrent infections. But the two other ends of the spectrum, which is autoimmunity, as a rheumatologist, I'm sure you would have come across a lot of such conditions, and something else, which is autoinflammation, which could clinically in some ways mimic autoimmunity, but characteristically different from each other. I'll give you a couple of examples. Now, this family was referred to us from the Christian Medical College in Vellore, and that was when my wife, who is a rheumatologist, was practicing at Christian Medical College. And this came to us with a very peculiar diagnosis of periodic fever. Now, with a professional diagnosis of familial Mediterranean fever, and the first thing that I said over phone was probably this is not familial Mediterranean fever because the disease doesn't occur in the country. Now, well, I was wrong, and like all wives are right, this turned out to be, in the case of familial Mediterranean fever, we did miss the mutation because As you see here, 2 mutations in a compound heterozygous state, just 1 nucleotide apart, could be missed in almost all standard exome or genome analysis methodologies. Now, this is actually a trivia to the entire problem. We believe now we have many more cases of such conditions where genetic mutations just 1 nucleotide apart can be missed in almost all the standard variant calling methods. We did go back and did Sanger sequencing for this entire family, and we did show that these 2 mutations segregate in the paternal and maternal lineage. And as you see here, so 4 is the index case. 4-5, who is the wife of this individual, is also a carrier. And so, of course, you could predict what's the chance for the kid to get the genetic disease. And as you see here, this is a consanguineous family. And family Mediterranean fever is something really interesting to look at, not just in terms of the diagnosis, but also in terms of ancestry, because as the name suggests, this is a disease very prevalent in the Middle Eastern population. So what we did is we could actually put this exome in perspective of the population genomes. And what we show here is that this is an admixed individual with part Persian ancestry and part Indian ancestry. And as the history goes, this individual's founder, many generations ago, around 54 generations ago, migrated from Egypt into India and formed this population in the eastern part of India. Now, that essentially extends us to a lot more populations in the Middle East and the West, where we were collaborating with the Middle Eastern populations. And what we also show is that this particular genetic variant in the MEFE gene is actually under natural selection in the Middle Eastern population. I know there are many more publications on MEFE and why it is selected in the population, mostly from Dan Kastner's lab, who is a doyen in the field of family Mediterranean fever. And that really comes us to a very interesting, what you call cultural and geographical heritage between Middle East and India. A lot of populations today that we find in India, they have been Indians, and they have been in the social fabric of India for many generations now, but nevertheless trace their ancestry from different parts of the Middle East. They came over ages as friends, as traders, as kings, so on and so forth. They have even contributed to, in many ways, prominent personalities in India over years, but nevertheless have a very small portion of the genome tracing their ancestry from the Middle East. And then therefore could also have, and in many ways have, genetic diseases which are otherwise prevalent in different parts of the world. And that's when we collaborated with a lot of countries to actually create a comprehensive resource for genetic variations in the Middle Eastern populations, involving a lot of countries in the Middle Eastern region, trying to not just understand their problem, but also being able to extend diseases from there into India, because we have probably a much larger population of individuals today encompassing close to 10 million in population size, who trace their ancestry from the Middle Eastern region. Now, what I've said all this time are cases which have come to the clinics and where clinicians have referred it to us. And this is essentially a bias in rare genetic conditions, or trace, as we call it, because not all patients do come to the clinician and seek medical attention. And that's especially true if the disease or the phenotype is associated with a taboo, or is not much of a functional implication in terms of mortality or morbidity. This is one such condition. This is hypogonadotropic hypogonadism. And this family incidentally was picked up by one of our collaborators, Dr. Sandeep Mathur in Jaipur. And we had a very hard time to trace this family and even draw a pedigree, because nobody would come forth, because other than the fact that they're sterile, they don't have other functional implications. So in many ways, we are severely undercounting the number of rare genetic conditions, because they don't really come to a clinician for medical care or medical attention. So that comes to the problem of how are you supposed to now find such conditions which don't go to a clinician? And the only way to do that is to essentially sequence normal individuals in their population. And the idea is very simple. Normal individuals could have been carriers of genetic diseases. And if you can estimate the carrier frequency in a particular population, especially if you have an endogamous community, you can sort of predict what is the prevalence of the disease, or rather incidence of the disease that would occur in the particular community. So we sort of did a pilot study on this, and this was part of a program called Indigen. And the idea of Indigen was to sequence around 1,000 individuals spread across the country and to address very, very pertinent questions to understand allele frequencies, to understand founder effects, and of course, how can we use that for public health applications. And the study actually revealed around 17 million absolutely new genetic variations which have previously not been described in literature or in any one of the large databases. Though there are significant overlaps with the global databases like the 1000 Genome and the Nomad, we still have a significant large number of genetic variants, many of them which are associated with genetic diseases, as we know from the patient samples. So the obvious real question is, can we now look at genetic diseases? Now, can we create maps across India, not just across India, but probably across the world, and now ask very pertinent questions on how to diagnose them? So this is really a top-down approach rather than getting the referrals right from the clinician. If you were to teach clinicians what are the conditions that we need to diagnose, we need to have an unbiased view of what are the genetic conditions which are out there. So the idea is very simple. You look at allele frequencies across this 1,000 genomes for genetic diseases which are out there. For this matter, let us put an example of autoinflammatory conditions. Most of the clinicians are not really aware about autoinflammatory conditions. So if you were to teach them what are the autoinflammatory conditions that we need to look for, or what are the clinical symptoms that you need to look for, can you ask from around 1,000 whole genomes and ask this question, are there frequent genetic variations in the population which are pathogenic as per the ACMG and ANP criteria? And if they're pathogenic and they're frequent, can we go back and ask clinicians whether they would see such conditions? And if they see such conditions, can we clinically correlate? Well, it's a quite ambitious program to start with, but I'll give you probably just one example to say that this really works. And this is essentially a map of all the autoinflammatory conditions and genetic diseases, genetic variants, which are prevalent and significantly different from the global populations. Just for convenience, let us mark three groups. One is ADA2, which causes DADA2. And for convenience and for the lack of time, let me just focus on another gene, which is MBK or mevalonate kinase, which causes hyper-IgD syndrome. Now, the mevalonate kinase variant has a frequency of around 7 in 1,000. So we expected that the number of patients which are diagnosed in India were severely undercounted. Before we did this, there are practically 0 cases of hyper-IgD reported from India. So we went back to the clinic. We looked at all the clinical symptoms of hyper-IgD reported in literature. And we sort of triaged every single patient who comes and matches 3 or more than 3 of these conditions. And we screened them for mevalonate kinase gene variants. We know that there are founder mutations. And what we found was remarkable. And what we found was that just 2 variants accounted for close to around 70% of all hyper-IgDs from that particular center. In other words, right from a genotype, you could now define the phenotype and you could screen for individuals who are eligible for that particular phenotype. And that is a practically new way of diagnosing genetic conditions. And in many ways, a very cost-effective way of diagnosing genetic conditions. Now, the application to this is, of course, you could do this over and over for many other conditions, including treatable conditions. And you could now offer genetic tests which are cost-effective, which are not genome sequencing, of course, but much more cost-effective genetic diagnosis, which can be used for not just screening, but also in many ways for family counseling as well as newborns. And today we have a chip which now does this, which encompasses around 10,000 odd genetic variations covering around 1,300 very prevalent genetic diseases in India. And this is now available, which is called Genique. Now, before I stop, let me take you through the entire circle. So you could start looking at patients and get referrals, and that is what I described as part of the Guardian Consortium. We could now educate clinicians on just not genetic diseases, but also provide them genetic tests which are cost-effective that you can roll out, not just in the hospitals, but also across diagnostic laboratories. By doing this over and over again, you could actually create awareness, and by creating awareness, you could screen more individuals. And at that point of time, you could hand it over to genetic diagnostic laboratories, which can now roll out across the country. And Of course, you could now identify genetic diseases which are previously never reported in the country, or in other words, which would never go to a clinician, or in many ways, the clinicians would not even understand that it's a genetic condition, by doing a top-down approach, by looking at population scale genomes, getting to the variant, and then asking the clinician to screen for very specific genetic variations and very specific genetic phenotypes. So in other words, what we think is we are moving towards what we call as P5 medicine, where we can predict a genetic disease. We can in many ways prevent a genetic disease already. We can make diagnosis and treatment very precise and personalized, but that's only possible if there's a lot of participation. And the central P of participation is the most important part of it. This involves not just clinicians, not just researchers, but a lot of groups, which include patient support groups, which includes cultural organizations, which includes religious leaders, who could now spread this awareness and advantages of genomics and can actually make a better future. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible. Almost all the next generation sequencing data was generated in the lab of Dr. Sridhar Sivasubbu, who's been a quite good collaborator for almost a decade and a half. Quite large number of clinical collaborators are part of the Guardian Consortium, and a lot of people who are in the lab and who were in the lab who have contributed to all these programs. And this is all possible with consistent funding. And today we are in the 4th round of funding from CSIR, who have consistently supported this program for over 15 years. I'll take questions. Thank you very much.
Thank you, Vinod. This is terrific. I would like to invite Dr. Igor Jurisica to start off the questions, and also the audience, please post your questions on the question answer tab on Zoom. Igor, would you like to begin?
Is Igor, do we have Igor
Yes,
online?
we
Or?
do.
Yes, we do. Sorry.
Igor, would you like to start the questions, please?
I was trying to look if there is anything on the chat, if anybody else is asking. Doesn't seem that we have any questions. So maybe one of the aspects relates to what we had earlier discussed in terms of the quality of data and databases. And so kind of consortia that are necessary globally across multiple countries to ensure that we have higher quality data, and going through existing databases, combining computational analysis, combining clinical and biological assays, in order to try to improve the quality of the data that we use for during these analyses. And so we had the discussion that in some diseases or in some platforms, it's easier because it's less competitive. So do you have any view on how to try to resolve it in more competitive or not as friendly areas to create this kind of global collaboration? And funding is obviously another issue.
So yeah, Igor, you raised a very, very pertinent question, and that is regarding the quality of databases and the quality of clinical annotations. Now, this is really a global problem. This is not a problem just restricted to one country or another. As genomic sequencing becomes commonplace, which increasingly is, the real roadblock has been and will increasingly be the quality of clinical annotations for the variants. Now, just to put in perspective, say a decade ago, what clinical genetics would typically be is that you have a patient with a particular genetic condition, you sequence that particular gene, and you find a non-synonymous or the missense variation. You typically annotate that as the pathogenic variant, and you publish a publication, right? And databases have actually curated this and said that this is a particular genetic variation associated with, say, disease X. Disease X could be any Mendelian condition. Now, when we see population genomes increasingly being done, especially from regions which have not been represented in global initiatives, like, for example, India or Southeast Asia for the Middle East, what you increasingly see is that such pathogenic variations are polymorphisms, right? Right. This typically is because a Middle Eastern individual would come to a Western country, to a large hospital, get diagnosed with a particular genetic condition, would be tested for that one particular gene, and that particular gene had a mutation, and just by association, and that person having the genetic condition, that mutation was marked as pathogenic, which is not really the causative mutation. Now, if you put a number to this, we did evaluate ClinVar very recently for all the variants that we found in indigen. And to surprise, what we found is 35% of genetic variations, which are marked in ClinVar, which is widely followed across the globe, are actually polymorphic variants, right? If you just take Indian and Middle Eastern and Southeast Asian genomes and ask this question, how many of them are polymorphic? There's a significant large number of variants which are polymorphic, which means that these are not the disease-causing variants. Now, fortunately, there are now common schemes for interpreting genetic variations, and one of the best known or best followed system is American College for Medical Genetics and Association for Molecular Pathologists. A quite elaborate scheme for evaluating evidence to ascertain whether a variant is pathogenic or non-pathogenic. Now, while this scheme is quite good and can be followed across the globe, the only issue is that then somebody needs to go back and reevaluate all the evidence. And that is really, really time consuming. And unless we have probably thousands of people contributing to not just evaluating the evidence, but also creating new evidence in terms of model systems, in terms of specific assays, we are not able to, at any point of time, solve this problem. And this problem is only going to grow and make it more messier, right? And this is essentially important because you cannot really tell a patient that you have this particular genetic variant, ClinVar says is pathogenic, and you might actually get a disease. Very important in cases, for example, BRCA1 and BRCA2, or for example, heteropolyposis colon, which are mandated to be disclosed to the patient. You cannot really have an intervention done on a variant which doesn't have much of evidence. Right? Just because a database says so, doesn't mean that the patient would have that particular genetic condition and should do that investigation. So yes, this is a global problem, and I'm sure if all people, including clinicians who see patients and phenotypes, could put together not just the genomic data, but also help in evaluating the evidence, this would really change the field.
Great. Just a follow-up question. Oh, you have one question here on the chat room. The question here, Vinod, is: do you foresee the creation of an India-wide biobank? And if so, what are the challenges you would anticipate?
Yeah, so very interesting question, and this is something that we have toiled for quite some years. And the sort of conclusion that we have arrived is, in India, actually patients are the biobank. So it's much easier to track patients and get the consent of the patients rather than keep their samples in a biobank.
I see.
It's much more cheaper, much more effective if you can get the consent of the patient to contact him back, right, rather than keep the sample at a point in time in your fridge.
I see. Okay, thank you. I had a question about that. That was a dramatic story, Vinod, about the retinoic acid for the ichthyosis. That was great. So my question really is about discovering actionable genes like that. How many circumstances where the discovery of the gene offers a new, not only an insight into pathogenesis, but an opportunity for treatment, like identifying druggable targets?
Right. So in most cases, you would not have a definitive therapy. When I say definitive therapy, that is a curative therapy. Like, for example, hematopoietic stem cell transplant will cure the condition, right? The patient doesn't need to take any more drugs.
Yeah.
Such conditions are extremely rare. I would say less than 5% of all the cases. But there is a large number of conditions which could be managed appropriately if you know what the genetic variant is, right? They're not curative in treatment, but they can actually make the life of the patient much better, right? And in many ways, these are investigational drugs, or these are drugs which are used off-label because there are enough evidence to support them, right? That numbers could be as high as 30% overall. But in very specific domains, I would say, for example, dermatology, I would say this number could be as high as 50%.
Yeah.
Right. So it's also domain specific problem.
Thank you.
So next question is from Dr. Amanda Ali. Thanks for your presentation. You mentioned your lab also looks at non-coding and circular RNAs. What are your thoughts on the clinical utility of these epigenetic mechanisms in the context of rare diseases? And do you think an unbiased identification, such as sequencing of these non-coding RNAs, is also a useful approach?
Yeah, so the quick answer is yes. At least if you talk about long non-coding RNAs and microRNAs, there are already genetic conditions, Mendelian genetic conditions, which are now associated with long non-coding RNAs. Now, your question is also, why haven't a lot of diseases been identified? The answer is, to identify such conditions, you would need to have whole genome sequencing. And clinical whole genome sequencing is only, probably a year or a year and a half old and not widely used across the world. So as people do more and more genome sequencing or graduate from exome sequencing or panel sequencing to whole genome sequencing, you will find a lot more genetic conditions which are because of non-coding RNAs.
Thank you.
I had a question, Vinod, about the genetic environment interaction in disease pathogenesis. I recall Dan Kastner talking about the MEFEVV1 gene modulating salmonella, for example, salmonella entry into target cells.
Right.
So my question really was, when you have a founder population, let's say you have a regional part of India that's quite, genetically intact and homogeneous, how do you, that could also be a population that is susceptible to a particular pathogen. I mean, a virus, chikungunya, maybe there's a virus in that population. So how does the geneticist actually address the environmental component in that circumstance?
Right. So there are two aspects to environment playing a part. The first aspect is what you sort of mentioned, where there is an environmental, bottleneck, which allows a founder population to survive, or founder genetic mediation to flourish in that particular population, which offers them some selective advantage. There are many examples for it. MEF is one example, sickle cell anemia is another example, which is widely discovered and reported in literature. The second aspect to it is individuals having the same genetic condition, right, manifesting a spectrum of phenotypes, right, modulated by the environment, right? And that's essentially something that is new and in many ways not really understood. And this is essentially where the regulation part, which is epigenetics, non-coding RNA, are all going to come into picture.
Right. Thank you.
Our next question is by Pratibha. Thank you, Dr. Vinod Scaria, for a comprehensive talk. Would you suggest exome sequencing or whole genome sequencing for detecting rare diseases? Right.
Yeah, so that's a very different question. It depends upon whom you ask and where you ask. So for example, if I ask a clinician in India where, for example, a whole genome or a whole exome sequencing is out-of-pocket expense for the patient, I would rather say, do your clinical workup, do your labs, arrive at a very specific diagnosis, and then do a very specific test, which is much cheaper, much more effective, because the expense is out of pocket from the patient. But in economies where, for example, insurance covers the cost, right, I could foresee increasingly whole genome sequencing would become a commonplace, not just for patients, but also for normal individuals. You have already seen that happening in many countries. Some of the Middle Eastern countries are doing population scale whole genome sequencing. That means every single individual or citizen of that country would get a whole genome sequence. Now, when you do it at scale for a very small population, this is an affordable proposition, right? But for a large country like India, where you're talking about 1 billion people, right, then of course whole genome sequencing for the entire population might not be something that is of the priority. But of course, as sequencing becomes cheaper, eventually, I believe a decade from now, increasingly a lot of normal individuals, including us in this meeting, would have our whole genome sequences done.
Thank you. Dr. Chandran.
Vinod, thank you. That was a great talk. I know you didn't talk much about it, but I'd like to highlight some of your work on the functional genomics. You had mentioned previously about your work using zebrafish. Could you talk about how the variants that you have discovered, you could look at the functional aspects of it in your lab?
Right. So just to give numbers, around 50% of the cases overall of the reference, we are able to give a diagnosis by doing a whole exome or whole genome sequencing. And this is just because the variant is there. There is a previous evidence on the variant, and there is enough evidence to say it is a pathogenic variant, so we can report it back. The next 50% of the cases, we don't have a clue. So typically what we do is we build in candidates, and the candidate variants are essentially sort of modeled or phenocopied in systems. And the systems could be cell-based models, depending upon the disease condition. This could be organ or organoid-based models in disease conditions. This could be whole organism models, like for example, zebrafish. We work extensively on zebrafish. And this depends upon the disease, the phenotype, and what you expect out of the model. So a lot of immunodeficiency conditions, for example, can be modeled in zebrafish because your readout could be the T and B cell populations, right? But for example, if you're going to ask a question about lung development, you cannot model in zebrafish because zebrafish doesn't have a lung, right? So it depends upon what are the endpoints that you're going to look for. A system, for example, if it's an enzymatic activity that you're going to measure, like for example, mevalonate kinase, just PBMCs from the patient is good enough to sort of understand whether the variant is pathogenic or not, right? You don't need to have complex model systems. But if you're talking about a brain development, right, and a gene involved in brain development or heart development for that matter, zebrafish is essentially a very fantastic model for that.
Dr. Juriska?
No more questions in Q&A. So maybe one other thing that we discussed earlier, but probably always worthwhile to engage more people in the discussion, is nomenclatures. So we talked about, for example, going into whole genome sequencing and then discovering a lot more microRNAs, non-coding RNAs in general. But one of the challenges is that some of them have been described earlier, have been put into the literature, put into the databases. Now the new ones are put into, and it's not only the annotation of which diseases or phenotypes they relate to, but also how do we refer them to. And so the whole aspects of how to ensure that proper naming is used and that we can actually somehow resolve the legacy problems that are already in the literature. And now we have been trying to do it for decades on genes and proteins. Now, long non-coding or non-coding RNAs is a newer phenomena, but it seems like we did not really learn from the past mistakes. So any thoughts on the direction of how to solve these problems going forward?
Yeah, good. It's a very, very pertinent problem. I think for the protein coding genes, people have sort of sorted this out, except for some small issues. Like, for example, you would have heard about Excel having problems with some set of genes. But except for those small problems, for protein coding genes, typically there is the human gene nomenclature, which is widely followed and is sort of a de facto standard. But when you talk about non-coding RNAs, I would say this is a real mess. I mean, a real mess of what do you call it, proportions that you cannot even really imagine. A couple of years ago, we just took one database of long non-coding RNAs. So we essentially took GenCode. GenCode is essentially a consortium which looks at the gene annotations. Ensemble puts these GenCode annotations online. We just compared versions of GenCode, say right from the first version to around the 36th version. And what we found was even within the same database, the nomenclatures have been dropped, they have been re-added, the same IDs have come up again, they're different genes, different genomic locations, right? And for non-coding RNAs, I mean, I believe this is like real mess. Somebody has to sit together and say that, I mean, let's have a rule and let's all follow this. Moving to more recent non-coding RNAs, for example, circular RNAs, right? Practically there is no nomenclature, right? People name it after their lab, probably their dog, I don't know, probably after their student, right? It's crazy.
I had a question, Vinod. The American Journal of Pathology just published something last week about the ethical implications of informing patients. So as we start to genome screen the population more, like when you find an unexpected genetic mutation, which may or may not be pathogenic, what are the ethical dimensions about informing patients about those discoveries?
Right. So there are only guidelines, there are no rules for that. And the best guideline that one could refer to is the ACMG, or the American College of Medical Genetics, where they have a guideline for reporting incident findings is that it should benefit the patient, and as usual, it should not harm the patient in any way. So diseases which are treatable, right? Yeah. And the patient could benefit from the knowledge of such a genetic variant. The general consensus is that the patient should know about it unless the patient has restricted himself out from knowing about it.
Right.
But theoretically, diseases which are not treatable, there is no point the patient knowing the fact unless he has asked for a particular information.
Right.
Right. Now, if a patient has asked for a particular information, he has to be revealed, irrespective of whether it benefits or not.
Thank you.
Any additional questions? We have time for one more question. Anything from the panelists?
Right. Vinod talked about all the people migrating in India over the centuries, and now you see the reverse phenomenon, people migrating out. We can see half of the panelists originally from your place. So any thoughts about, looking at families migrating here? And we take, for example, Canada, you have 350, 400,000 people coming every year, or half of them are from South Asia, so right? So any opportunities for collaboration?
Absolutely. So this is something that we are now currently exploring. To just give a short history of what we have just been doing. We just published last week a publication on the SAMD12 repeat expansion in a very specific community in South India. This is a small endogamous population in South India. And over the last, I would say, 300 years, members of this community have migrated into Southeast Asia, Australia, Malaysia, Singapore, so on and so forth, Sri Lanka, for example. And we have the same disease in all these populations, right? Now it is very important to now trace your ancestry, right? And also be able to say that if you have this symptom, this is the mutation that you should look for, right? That makes the life much easier for the patient, the community, and their family, because they can be aware about the disease, right? And especially in a new country, for example, Australia, right? Clinicians might not even recognize the condition, which otherwise is very prevalent in India, right? So yes, this would indeed be a great source of or an opportunity to collaborate across, because the Indian diaspora, or rather South Asian diaspora, is now across the world. Canada is a great example. The United States is another example. Middle East, we already work with them. So we would be more than happy to extend this. And the idea of making the indigent data publicly available is that clinicians across these countries should also be able to now search the Indian frequencies, right? So that they can make a better job.
Thank you, Dr. Scaria. Exceptional talk, and thank you for taking our time to speak today. And also to our panelists, Dr. Chandran and Dr. Jirasika. To the audience, thank you for joining us today. Please join us on January 26th for another lecture. That's going to be our lecture number 20 by Dr. Anne-Marie Malfayet on pain and arthritis, origin and pathology. See you all on January 26th. Thank you again, Dr. Scaria. Bye-bye.